And I will introduce to the first speaker, who is Adolfo García-Sastre. Adolfo García-Sastre is a professor of medicine and microbiology. He's a co-director of the Global Health and Emerging Pathogens Institute of the Icahn School of Medicine at Mount Sinai Hospital in New York. He's a professor in the Department of Microbiology at Mount Sinai University. His interest in research has focused in the biology of RNA viruses such as influenza or coronavirus, and he's a world authority on this matter. He's Spanish, he's from Castilla, born in the city of Burgos, where he received the honoris causa doctorate in 2018. It's a privilege for us to have Professor García-Sastre, who will discuss the topic of the interaction between the virus and the host.
Hi, hello, good morning. I hope you all hearing me well. Pleasure to be here. I would prefer to be in Madrid, but you know, travel restrictions don't allow us to travel so quickly. I put at least as a background in my Zoom Burgos. So you can imagine that I'm in Burgos, although actually I'm in New York. Okay, so I'm gonna be talking, I guess I can get, I don't know if I can get, yeah, start. Okay, I think. So I'm going to be talking about our approaches that we have about looking to the interactions of SARS coronavirus with the host and how we can use the knowledge of these interactions into looking into potential antivirals that can be developed quickly for the treatment of COVID-19. And my first slide has to do actually not with SARS coronavirus 2, but with influenza. The reason is because most of my research has been concentrated in influenza. And we are now using the tools that we have developed to understand influenza now to study SARS coronavirus 2, applying the same tools but trying to do it in a more accelerated manner in order to come out with potential therapies that may be useful for the treatment of this pandemic disease. So just to illustrate here that the influenza virus is a negative strand RNA virus that encodes around 10 proteins. And in the next slide, we see the coronavirus is also an RNA virus, but is one of the coronavirus are the largest RNA viruses that we know, and instead of encoding around 10 proteins, which is what is the average of proteins that encode different RNA viruses, this virus encode 3 times more proteins. It's because it has a very large RNA genome, and these proteins are the majority expressed as a part of a large polyprotein that is being cleaved into a total of 16 different NSP proteins, non-structural proteins, but in addition also encode several structural proteins, 4 structural proteins, and some other accessory factors that are not encoded by the polyprotein, that are encoded as subgenomic RNAs, that also they are non-structural proteins that interact with the host and manipulate the host in order to facilitate viral replication. Now, in terms of influenza virus, what we have been looking is that as any virus, when it enters the host and it replicates, is using a set of host factors that needs to identify that are required for the replication of this virus. In addition, the virus interacts with antiviral factors that are being induced inside the cell trying to prevent viral infection. And the virus is trying to prevent the action of these antiviral factors. And it's these interactions between viral factors and host factors, either require host factors or antiviral factors, what at the end determines the outcome of the replication of the virus. The virus needs to use these factors or to counteract these factors in order to be able to replicate. And it does so through the genes that it encodes. The same thing is happening with coronaviruses. Coronaviruses, they have a different replication mode. They replicate in the cytoplasm instead of in the nucleus like influenza, but they also have required host factors that are required for replication and that the virus needs to identify and to interact with them, as well as antiviral factors that the virus needs to counteract in order to be able to replicate. So the question that has been asked in my lab and in many labs for many years is which host factors are important for virus replication and pathogenesis. And in terms of influenza, we have a large consortium through a U19 NIH funded grant that is using systems biology approaches in order to identify these host factors that are required for viral replication, with the concept that if we know these host factors, we also may know new strategies how to inhibit the activity of the host factors that are required in order to inhibit viral replication. And this is one example of some of these things that we have done with influenza. What we see here is an interactome map in which in yellow, they are the different influenza virus proteins, a total of 10 proteins. And in red, there are host factors that are known to interact in an interaction mapping done by proteomics in collaboration with Nevan Krogan. But not only that, which ones of these host factors interact with the viral proteins, that they make an impact in viral replication when you silence these factors. And through that, we have a rich map now of around 80 host factors that we think they are very important in order to regulate influenza virus replication. And some of these host factors we have been targeted with known inhibitors like 661 with CT8 or topoisomerase 1 with camptothecin, and some that actually these factors are able to inhibit either disease or inhibit viral replication, disease in animal models at this moment. So we are using now a similar approach for COVID-19, for SARS coronavirus 2. And what we are doing in terms for looking for a therapy, we have heard already from Dr. Cliff that there are approved factors that are used for Dr. Cliff Lane, that there are factors that are approved for emergency use at this moment, like remdesivir, that plasma from convalescent individuals, like dexamethasone. But we want to identify more approaches and perhaps some that may be even more successful. And for that, one approach that we and many other labs are using is concentrating in drugs that have been already being used in humans, either in clinical trials, or they have been using for another disease. That's what is called drug repurposing. And what we try to do here in our approach is to identify, based on predictions based on omics studies, host factors that are known to be targeted by some of the drugs that we know how they behave in humans in terms of PK and in terms of side adverse events, because they are using already in clinical trials or because they are using for another indication. So basically, drugs that are already used in humans, based on predictions based on omics studies, we look for inhibition of viral replication in tissue culture. And the ones that give good impact, we are trying to move them into animal models or sometimes directly into clinical studies. And that's what I'm going to be talking about in the next slides. So again, here the question is, can we identify compounds that inhibit host factors involved in SARS coronavirus 2 replication among known human drugs? And one of the first things is, what host factors are involved in SARS coronavirus 2 replication? So one way why we're doing that is by looking the proteins, the host proteins that the viral proteins interact with the idea that if a viral protein is interacting with the host protein, probably this interaction is important and this host factor is important for the viral replication, either because it's an antiviral factor that it needs to be counteracted by the virus, or because it's a proviral factor the virus is going to be used for its own enhancement of viral replication. So we are collaborating with Nevan Krogan, which here in this picture. So basically what Nevan has done and his group, he took all the different proteins encoded by SARS coronavirus 2, expressed them individually in cells, in human cells, pull them down, and then find out by mass spec what human host factors are interacting with these different proteins from SARS coronavirus 2. And with that, he came up with a map of protein-protein interactions with 332 protein-protein interactions between viral proteins and host factors. And from that, in collaboration with some chemical bioinformaticians, we have selected a total of 69 druggable host factors that they are known drugs that are able to interact with these host factors that interact with the viral proteins. So this is these predictions have been done by Kevin and Brian, which are also part of this big group of people that we are working together. And again, here there are some of the factors that are interacting with the viral proteins on the right of the screen, and some of the compounds that are targeting some of these factors. So we are now knowing these things in our lab. Chris and Lisa, and later on also with Elena and Romel, we have been looking to the activity of these drugs that are known to antagonize a particular host factor, interact with the viral protein, whether they make an impact in viral replication. And for that, we developed a medium throughput viral replication inhibition assay that looks for antiviral activity of these drugs, in which we seed cells that are permissive for replication of the virus. We started with Viro6 monkey cells because they replicate the virus very well, but now we're also moving into human cells just to confirm the data that we get also in the Viro cell, in the Viro6 cells, and some of them are also primary systems for replication. So we seed these cells into 96 well plates, and then we treat them with the different drugs at different concentrations for 2 hours. We add the virus, we lead the drug with the cells, and with the virus, we add the virus at an MOI of around 0.025. We are conducting redundant studies with another group in Paris, in the Institut Pasteur, in order to contrast results. And then 48 hours later, we fix the cells, we inactivate the virus, we take the place out of the BSL-3 because now the virus has been inactivated, and then we measure the amount of infection that we have gotten in these cells in the presence and in the absence of a potential inhibitor. And we do that by staining against the nucleoprotein of the virus to see what is the number of infected cells. But we also conduct assays on viral replication on the supernatants for these compounds that are working well. And similar things have been done in the Institut Pasteur. Is that the MOI is slightly different, and they look for viral replication by RT-PCR, not by staining of MP. But the results that we got, they are very comparable. All right, so this is, for example, how they look like, the plaques that the virus induces in cells. So we take the supernatant of these cells that are being treated with compounds in the presence of the virus, and then we look for the viral titer by plaque assay in new cells. So how does it look, for example, a known inhibitor in this assay, like remdesivir? So this, what we assay in these assays is we take a particular drug, we use it at 6 different concentrations, from high concentration micromolar to concentrations that are close to nanomolar. And then we look for viral replication. You see here in the solid line that in Viro cells, remdesivir inhibits well viral replication with an IC19 inhibitory concentration that inhibits 90% of viral replication. Of micromolar, it actually works even better in human cells because the uptake of the drug is better in human cells. And what we see in this line that is not solid line, we see also we do at the same time a cytotoxic assay to make sure or to know whether the drug is also killing or not the cells. So in this case, remdesivir is not cytotoxic, and the cells are perfectly happy in the presence of remdesivir. But when you add the virus, the remdesivir is able to inhibit viral replication, and it has been described for remdesivir. Now, so I mentioned that in human cells, remdesivir is working better, and then when we use 293 T cells, which are human cells that they are needed to express AC2 for this assay, because 293 T cells don't express the receptor, so we make them express the receptor. Now the IC90 goes from micromolar to around nanomolar range, around 20 nanomolar range. So that's a very good drug that is now has been approved, and I think it will be even work better if you use it early on during infection. The only problem is remdesivir is being given intravenously, so it's very difficult to use as a general treatment for everybody that gets mild symptoms. All right, so with this antiviral assay, now we have conducted assays for all of these drugs that they were predicted. We have tested now more than 100 compounds, either coming from this study or from another studies, and some of them, they appear to have very nice antiviral activity. So these are the samples of the first compounds that we found. We found 10 agents that showed good efficacy in killing the virus, in reducing viral replication. So again, consistent with the data that we have with the Institut Pasteur. And among them, there are some factors that inhibit protein translation, like sotatifin and ternatin and plitidepsin, and some of the factors that are targeting sigma-1 and sigma-2 modulators, which sigma-1 and sigma-2 host factors, and compounds that modulate the activity of these and that target these sigma-1 and sigma-2, which are receptors that are not very well understood, but they have multiple drugs that target these receptors, including, for example, hydroxychloroquine, but also some of the drugs that are being used for different indications, as indicated here. Actually, of these drugs, the one that seems to be working better in our hands is PB28, and is the one that we are exploring further. So these are some examples with these drugs that bind and inhibit sigma 1 or 2. All of them are able to inhibit viral replication here in red. With PB22 seems to be one good one. These sigma 1 and sigma 2 are able to interact with two of the proteins of the virus, NSP6 and OF9C. And again, there are multiple drugs for multiple indications. The one that gives us better antiviral activity at this moment is this PB28. And if we compare, for example, PB28 for binding to sigma receptors versus another receptor that is very similar, which is human ERK, that is believed to be the one causative of the site adverse events of hydroxychloroquine, PB28 is able to bind much better to sigma 1 and sigma 2 than to human ERK, but hydroxychloroquine binds identical to human ERK in these binding assays than to sigma 1 and sigma 2. So just from these predictions from in vitro experiments, we will predict that perhaps PB28 has less potential side adverse events than hydroxychloroquine because it does not target this receptor that is in the heart that is believed for some of the problems, cardiovascular problems that hydroxychloroquine may cause. And if we compare the Activa activity of hydroxychloroquine with PB28, actually it's more potent. So hydroxychloroquine in tissue culture has an IC90 around micromolar, but PB28 is sub-micromolar, 20 times more active. So we think that this is a good place where we can start to look for inhibitors that are better than hydroxychloroquine and that target also sigma receptors. I also mentioned some of the translational inhibitors. So that I think we use it because it targets EYF4A that we found that interacts with one of the viral proteins. And this has an IC90 quite good, around is again in the nanomolar range. The non-solid line shows what it appears to be some cytotoxicity. So you get 50% in terms of live cells in the presence of sototafin. But this is not because sototafin is killing cells, it's because sototafin is inhibiting the cells from dividing in these 48 hours assay. So in the absence of the drug, the cells double. In the presence of the drug, the cells don't divide, so even doesn't kill cells, we get still 50% less cells at the end of the experiment in the presence of sototafin than in the absence. So we think that despite this apparent cytotoxicity, these compounds may be used for the treatment in vivo. And in fact, some of these are being used for other indications in humans. And one of the compounds that we found that has higher activity is plitidepsin. In the beginning, we used ternatine, but plitidepsin is another inhibitor of EYF1A that was described already as an inhibitor of SARS coronavirus, but in this case, now we use SARS coronavirus 2. And in the case of this compound, which by the way is being tried to be developed by PharmaMar as an inhibitor of SARS coronavirus 2. PharmaMar developed it as an inhibitor of myeloma and has been able to license it and is being used in several countries for this indication. Plitidepsin that targets EYF1A, that this is a structure of the EYF1A bound to plitidepsin, has an antiviral activity which is in the range of very close to nanomolar, which is much better than the remdesivir, not only in viral cells, but also even in human cells. Remdesivir is around 20 nanomolar in human cells, and still plitidepsin is around 1 nanomolar in human cells. So we think that this may have, and it's good that it's being used right now in clinical trials because we think it's one that has a lot of potential. So this is now the inhibition in human cells, and in human cells, aplidin or plitidepsin, which is approved in Australia for the use in multiple myeloma, again has a very good IC90 that is sub-nanomolar. So again, we think that this could be a great inhibitor. Okay, and phase 1 clinical trials for SARS-CoV-2 have already begun with this drug, and we are now testing them in animals. Another way how to look for inhibitors is looking to proteins that become phosphorylated during viral infection. And that's another study that we have done in collaboration with Nevan Krogan and Jeff Jobson, in which we look not now for the factors that interact with viral proteins, but for factors that become phosphorylated in the presence of infection, with the idea that if they become phosphorylated, again, this is because it could be used for the advantage of the virus. So this is a phosphoproteome map in cells that are infected, and these are predicted kinases that are able to phosphorylate these phosphorylated proteins that we see during viral infection. And now, based on these predicted kinases, then we look to inhibitors of these kinases where they're able to make an impact also in viral replication. Non-inhibitors. And among them, we have inhibitors of PIK5 Apilimot
y también con los inhibidores del PK5.
Yeah, Apilimot is a good
Apilimot
inhibitor.
Es un buen inhibidor.
And I don't know, I hear now an echo in English of myself, so I don't know why is that, but the echo just finished. Okay, Apilimot seems to be a great inhibitor, but also inhibitors of CDKs seems to be also good inhibitors of viral replication, and inhibitors of the MAP kinase pathway, especially the one that involves MAP kinase 3 and MAPK12 and 13 and these other MAP kinases that are able to inhibit viral replication. Also, when you use an siRNA against these kinases, then you also see inhibition of viral replication, confirming that these kinases are important for viral replication. And the last kinase inhibitors, and some of them are FDA approved, so that's good, or they have been used already in humans. And the last kinase inhibitor I'm going to show you is an inhibitor of kinase 2, which seems to be required for an interaction with the end protein of the virus for viral aggression. And it's again also a relatively good inhibitor of in vivo E6. All right, now I'm going to just show a few more studies very quickly about some of the approaches that we are using with other people. This is a collaboration that we have with Gaetano Monteleone and Bob Crook, that they have been looking to potential repurposing of hepatitis C virus proteases inhibitors, because they resemble a little bit the protease of hepatitis C to the main protease of SARS coronavirus 2 in terms of the structure of the active site. So some of them are predicted also to inhibit the protease of SARS coronavirus 2. And among some of these protease inhibitors, although working on activities that are micromolar in terms of inhibition, boceprevir and grasoprevir are able to inhibit, as well as cimeprevir and baniprevir. But again, they are high micromolar concentrations, but they could be also a way how we can start to think about how to improve some of these protease inhibitors. Now, the interesting thing is that if we use a combination of remdesivir with grasoprevir, we get synergy, meaning that in the presence of remdesivir, at a concentration that inhibits around 50% of viral replication, we require less grasoprevir that we require to inhibit even further viral replication. So they synergize, and this can be seen in a curve that is this one, in which we see that the combination of grasoprevir and remdesivir allows for less use of remdesivir and less use of concentration of grasoprevir than the ones that they are required, they are using individually. So this may allow actually combination therapy to be able to use some of these protease inhibitors together with remdesivir to be able to make an impact in viral replication. And we assume that the reason why they are synergistic is because the protease will inhibit processing of the polyprotein, the inhibitor of the protease, processing of the polyprotein that gives rise to the replicase, and the replicase is the target of remdesivir. So in the presence of the protease inhibitor, we predict that there is less produced polymerase, and then you require less remdesivir in order to inhibit the remdesivir level, the polymerase levels that are lower in the presence of remdesivir. All right, and so in summary, the SARS coronavirus interactome and phosphoproteome provides with rational therapeutic targets for drug repurposing. We have identified inhibitors of sigma receptors of translation coronavirus 2 replication in vitro. Some of the hep C protease inhibitors inhibit SARS coronavirus 2 at micromolar concentrations, but interestingly, they synergize with remdesivir. But obviously, further clinical and preclinical studies in animal models are warranted. And I think that the combination therapies is something that we should consider for the treatment of this virus, not only because they may synergize and may allow the use of these inhibitors at lower concentration that perhaps allows for more widely use or for less side adverse events, but also because it may reduce also the potential of resistance of these appearance of resistant mutations that resist the action of some of these antivirals. And with that, I just gonna acknowledge this is my lab, but I've been collaborating with many other people that I think have been mentioned during my presentations, and also funding coming from different agencies, including NIH, Department of Defense, DARPA, and several philanthropy agencies. So I will stop here and I will take questions if we still have a little bit of time.
Thank you very much, Dr. Garcia-Sastre. Unfortunately, we're very tight with time, so we're going to move on to the next panel, but we will keep the questions. If anybody wants to ask questions, please remember that you have a form right underneath the screen where you can write who you want to address the question